And then let's go to the slide deck. There it is. Okay. So for today, we're going to talk about gram-negative and gram-positive cocci of medical importance. So the stuff that's that those of you going into the medical field will probably come across, or some things that you may have already come across personally, directly or indirectly, or just things we've all heard of. So hopefully this will not be esoteric, and we'll be kind of getting into the meat of microbiology. This is the outline of everything we're going to talk about in this lecture. Okay, so we break down cocci bacteria, and remember, cocci is the circular shaped bacterial cell. And we break it down into staphylo, meaning clusters, and strepto, meaning strips or lines or tracts. And the vast majority of these cocci bacteria are gram positive, with the exception of a couple Neisserii genera that are gram negative. Okay. And so, as we go through these, you'll need to be familiar with these different flavors. You also should be familiar with these general characteristics of Staphylococci bacteria and how they are similar and different to Streptococci. All right. There's going to be a lot of crossover, but there are going to be some differences. So, for instance, in general, Staphylococci are going to be on the skin and in the mucosal membranes. So our nose, our mouth, our intestinal tracts. Again, because it's cocci, they're going to be circular or spherical cells arranged in these clusters, these great. Oh, sorry. Someone calls me and it just gets rid of my screen. Okay. And like I said, the majority of these are actually, all of the Staphylococci that we'll talk about are gram positive. They don't have endospores, they don't have flagella, and that's usually because they are in these clustered like formations. So flagella and endospores are more typically found on single cells. It would be really hard for a cluster of cells to all move in unison, doing their different flagellar movements in coordination. So typically these are going to lack those structures, but they do have capsules in some cases. Remember, the capsule is that thick outer coating to protect it. And there's a large representation of medically relevant species in the Staphylococci flavor. The big one is Staphylococcus aureus, or Staph aureus. Okay. These are going to grow in these large colonies. Let's annotate here. So we're going to have these nice large colonies. They're kind of opaque. Their optimum temperature is 37 degrees, right? Because that's body temperature. So that's going to be the temperature that they thrive in, which means that they do very well growing inside us. Now, it's their optimum temperature. It's not their required temperature. They can also survive at room temperature. So that's why they can linger on our skin, waiting for something like a wound to occur to then gain entry inside. They're tolerant to high concentrations of salt, different pH extremes, as well as high temperatures, which makes them a very resilient germ. On top of that, they have lots of virulence factors that we'll get into in a little bit. And one of them is an alpha toxin. So the alpha toxin here is what it secretes. And so what that does is that breaks down the hemoglobin in a way that's called beta hemolysis. There's beta and alpha. We'll go into those in a little bit. But essentially, what that means is if you grow this Staph aureus on a agar plate that has blood in it, so it's a blood agar plate, just like hopefully some of you see these zones of inhibition where the antiseptics or the disinfectants exclude the growth of bacteria, what we see is a zone of hemolysis. So it's breaking down the hemoglobin in the plate. And so where you see the bacteria, you see this zone around it because it's secreting that alpha toxin to break down the hemoglobin. So Staph aureus can be found pretty much wherever humans are. It's readily isolated from fomites. Now, we talked about fomites several weeks ago. Fomites are anything that bacteria can survive on that are frequently touched by us, right? So doorknobs are a fomite. Toilet flushing levers are good fomites. Things that we come into contact with our hands, and then it seeds our hands, and then we, absentmindedly touch ourselves and then spread that around to other places. The carriage rate, or just how many people have Staph aureus on them in healthy adults, is 20 to 60%. So there are a lot of us in this Zoom meeting right now who have Staph aureus on us, but luckily it's not infecting us at this point. Okay. Typically, where we have it is going to be the anterior nares, or the outside of our nostrils, our skin, the nasopharynx, and in our intestine. And if you are living in conditions where hygiene is low and nutrition is poor, and you have sores or wounds on the outside of your body, you are going to be much more likely to have an infection. Okay, there's a difference between just carrying it and being infected with it, right? If you have pre-existing infections, then you could become co-infected with this because your immune system is busy on the fronts fighting some other infection. Or if, of course, you're immunodeficient, you're also going to be more likely to get an infection of staph. And the problem with staph aureus is that this is the one that is the methicillin resistant Staph aureus, or MRSA. Okay, so we're finding, particularly in hospitals, that this is lurking, and we can't treat it with our normal assault of different antibiotics. This can range from a localized infection, right? So just at the site of a wound, but it can go to systemic. And this is talking about staphylococcus in general, not just Staph aureus. Okay, so now we're going into just generally speaking, disease from staphylococcal infections can be things like folliculitis, which is a localized infection near a hair follicle. Okay, usually you don't need to treat it, it just resolves by itself. However, if that continues to progress, we get a manifestation called furuncle, which is a fun word to say. That is the boil that we're familiar with. That is much more inflammation from your immune cells. You have your sebaceous glands that are producing sebum over time and leading to an abscess or a pustule. If it progresses from there, it can get into a carbuncle, which is equally fun to say. And this is going to be now much less superficial. It's going to penetrate deeper into the skin and become subdermal. Um, and now you have essentially an aggregation of furuncles. And these are the things that that are posted on YouTube that you either like to watch or you can't stand to watch. I personally am not of that kind of persuasion. It grosses me out. Um, but they call them popping pimples, right? But these are not pimples. These are things that are well beyond your typical zit. Um, and then also impetigo. Impetigo is not a carbuncle, progressing. It's a different type of skin uh, infection, and you get these bubble-like blisters that will peel away and break. And it's usually a newborn infant child um, disease.
Hey, Carl.
Yes.
So the staphylococcal disease, is that only pertaining to Staph aureus?
No.
Is that's like the all of staph infections react that way?
They don't all react that way, but this is the range of which they can react. So this is not just pertaining to Staph aureus. This is staphylococcal disease in general. It can be anything from localized to systemic, depending on the type of staphylococcal disease. And we'll go into more resolution on that as we go through the slide deck.
Thank you.
Yep. So Staph aureus, this is looking at a cross section of the skin. And here we see that there is the hair follicle. And let's get to my drawing. All right, here's the hair follicle right here. And well, that's the hair, but the hair follicles down here. And then what happens is the bacteria gets down into that follicle and starts dividing and multiplying. Okay. And then that signals to the body that there is bacteria, there are foreign invaders. And so what do you get? You get a bunch of infiltrating granulocytes. Remember those? The phagocytes. You get macrophages come in and they start trying to chew up this bacteria. And then what becomes problematic is that if you also remember when we were talking about how the body responds to microbial invasion, it starts building this wall of fibrin around that whole infected site. It's good because it walls off the invaders, it prevents them from spreading. But what happens is by walling that off, if the bacteria isn't completely controlled, now it will just continue to divide. And then you have this pus, which pus is mainly dead uh, immune cells and then dead bacterial cells. But it kind of becomes walled off and then it just continues to grow, right? It can't release that pus. And that's how it goes from, like a simple boil to more like a carbuncle. Right? And then you get these huge abscesses. So it's again the body's attempt to prevent systemic infection, but leads to these really nasty sores. Here's some pictures of some folliculitis caused by Staph aureus. So kind of like almost looks like a rash um, broadly across the skin. When that rash gets to be worse, forming an abscess, it looks more like this. So a red inflamed bump with all of these pustule pockets surrounding it. That's a more localized infection, but also a much more troublesome infection. Other manifestations of staphylococcal disease can be osteomyelitis. So that is when you have a, a staph bacteria that has gone into the bloodstream. So now we have a um, a bacteremia, right? Not necessarily a septicemia yet at this point, but it's bacteria that is now in the blood system, and this can be introduced during surgery if the medical devices weren't completely decontaminated. And it's particularly bad because once it goes into the circulatory system, now it can reach the heart. And that is when you get things like endocarditis. And that's very, very life threatening. But with the case of osteomyelitis, that is a very debilitating disease and a hard disease to treat because remember, the circulatory system goes to the bone. Osteo is bone. And so what's happening is these staphylococcal bacteria are entering into the um, into the site of the bone and taking up residence there, and it can be very challenging to treat. And if you, as you can imagine, if you had an abscess forming in your bone, that could be very painful. There are things like food intoxication. So you eat the contaminated food, but it's not the bacteria. Because remember, we talked about food intoxication. It's not the bacteria that make you sick, it's the toxins that make you sick, the toxins that they secrete. And so, if they happen to be heat stable, even if you cook your food, they are still present in that food. And then that leads to um, different GI discomfort. There's scalded skin syndrome. Um, and that is going to be in this picture above where it looks like a burn. Essentially, it's a blister and a desquamation of the epidermis. That just means that the cells, the outer layer of cells, no longer adhere to the cells below them, and they start, sloughing off. You can also have something in the picture below called toxic shock syndrome. No, I'm sorry, scalded skin syndrome is also this one below. This is an infant who has it, and basically the entire outer epithelial layer of the cells have blistered off, and the cell junctions, essentially they attack the desmosomes, which if you've had cell biology, the desmosomes are important for connecting cells together and holding them together. They come apart and now the cells aren't connected to anything and they just slough off. Toxic shock syndrome is just going to be um, the toxemia, which is essentially all the toxins from these bacteria that they secrete build up and become systemic. And then once they become systemic, they start traveling to your different organs and can lead to organ failure. Other things to mention, we have coagulase negative staphylococci. That's just coagulase is a test. So if you want to try and identify a certain bacterial species with the 6 eyes that we talked about at the beginning of this course, then you will, one of the things that you can do is determine if it's coagulase positive or coagulase negative, meaning does it coagulate a certain media that you put the bacteria in. And so the coagulase negative staphylococci are going to be a large representation of some of the healthcare associated and opportunistic infections that arise in immunocompromised patients. So, those nosocomial infections that we talked about, right? Hospital acquired infections. Um, are going, a lot of them are going to be these coagulase negative staphylococci. They're opportunistic, meaning they take advantage of a weakened immune system, and that's why they thrive in hospitals where people are sick. Right? Not as big of an issue for people that are immunocompetent and healthy. And these are going to be things like S. epidermidis, because it's on the skin. However, it can also go further into the mucosal membranes, cause endocarditis or inflammation of the heart, bacteremia, systemic, or urinary tract infections. We actually use a different strain of epidermidis in the lab. It's not virulent. So they can range from being opportunistic to being pretty benign and safe. That doesn't mean you don't want to take precautions, but to give you an idea, had we had a face-to-face lab this quarter, this is one of the bacterial species that we would have worked with at some point. And then S. hominis, because it infects humans, and it lives around the apocrine sweat glands. So that's going to be a gland kind of on like near hair follicles and on the surface of your skin that secrete sweat. And so once it clogs those pores, you can get acne and things like that. S. capitis is going to be on the scalp, face, and external ears. So that primarily lives on your head, hence capitis, right? So all of these names, all of these species names can help you if you're learning these, because they're very clear on what they do. So if I had these three and I said, list the primary uh, tissue that these different bacteria thrive on, you could use the names to help you, right? Um, what about S. saprophyticus? That's on the skin, intestine, elsewhere. What do you think? What does that name tell you? I know you've heard saprophyte before, right? Nobody remembers what a saprophyte is? Okay. Well,
Doesn't it have to do with fungus?
yes. What do fungi do typically to get their nutrients?
Decompose stuff.
Decompose. Okay. So saprophyticus is going to secrete enzymes that decomposes the surrounding tissues, leads to necrosis. So why are these important in a clinical setting, aka in a hospital or a medical facility? Because the vast majority of them, 95%, have penicillinase. Penicillinase is the enzyme that breaks down penicillin and ampicillin. So these antibiotics have no effect on them. And if you remember, we talked about strategies that bacteria have evolved. When we talked about evolving antibiotic resistance, one of the strategies was to have enzymes that break down the structure, that beta lactam ring, to then make that antibiotic functionless. As I mentioned before, MRSA is a huge issue in hospitals and continuing to be on the rise and threaten a lot of lives. Some strains of MRSA are resistant to every single major drug category, with the exception of vancomycin, and it's only a matter of time, really, before it evolves a workaround to vancomycin. Oftentimes, these abscesses have to be lanced so that they can drain. Because remember, the fibrin walls them off and they have nowhere to go. It's good that they don't go systemically, but they can't get out either. And so you have to go and have the boil perforated. And again, some people love, my wife is a physician. She loves doing that stuff. Yeah, not for me. But and then also this can lead to a very lengthy treatment. So remember when we talked about the microbiome, right? If you have a staph infection and you have to undergo a long period of intensive antibiotic treatment, what is that doing for your healthy gut flora, right? It's trashing it. And so as a result, you can get opportunistic pathogens come in and take up residence. And so it's something that is quite quite the problem in this day and age, and is only going to get worse unless we find some new approaches. All right, that's Staphylococci. Any questions or comments on that before we move into strepto? Nope. Okay. Remember, strip chain, that's the formation of these coccobacteria. And the general characteristics of strepto are going to overlap to some degree with staphylo, but in some cases, they'll be different as well. So again, they're going to be oval or circular. They're going to be chains, sometimes in pairs. What is a pair? What are two? When there's just two cocci cells stuck together, what do we call that?
Dicocci?
Close. Diplococcal. Non-spore forming, non-motile. So again, these also don't have flagella, right? Because they're these long chains. It would be too difficult for these long chains to all move in a coordinated fashion. They can form capsules and slime layers, just like the staphylococci. They're facultative anaerobes. Okay, so that means they can live in an anaerobic environment without oxygen or low levels of oxygen, but they can also exist in oxygen in the presence. So that makes them particularly bad because they can live on the surface of your skin in a highly oxygenated environment. And then they can penetrate into tissues that have less oxygen. And so they can thrive in both settings. And so that makes them just on the ready, on your skin, waiting for some injury to allow entrance. What else? They don't form these catalase enzymes, which we'll talk about when we get into beta and alpha hemolase, or beta and alpha hemolytic systems, but they can use peroxidase. Why would peroxidase be beneficial? What does it do? What do you think it does?
Doesn't it break down hydrogen peroxide?
Yes. Why is that good for bacteria?
For their survival?
Yeah, right. We said that hydrogen peroxide is a germicide, right? Because it attacks biomolecules, because it's a free radical. And so if you are encountering hydrogen peroxide, but you can, or other peroxides, other radical oxygen species, then you've got your self-defense. Okay. They're sensitive to drying, heat, and disinfectants, luckily. Okay, so these are going to be things that are less likely to cause nosocomial infections, going to be less likely to be transferred via surgical equipment, right? Because they are less resilient to the treatments that we give surgical equipment. But different subgroups. So we have beta hemolytic and alpha hemolytic. Both of them are hemolytic, meaning they break down hemoglobin. The difference is that beta completely breaks it down, whereas alpha hemolytic partially breaks it down. Okay. And so if you were to grow these on a blood agar plate, what you would see is that beta hemolytic streptococcal bacteria will have that zone of hemolysis, which is much more defined. Okay, alpha hemolytic, it's harder to detect because it doesn't break it down as efficiently. And what is governing that is the different toxins that a particular species has. So with beta hemolytic, you have this exotoxin called streptolysin. Okay. Strepto because it's streptococcal, and lysin because it lyses the hemoglobin cells, right? The blood cells. It doesn't lyse the hemoglobin per se, it lyses the red blood cells. And so what the beta hemolytic category is going to be sensitive to is certain medications called bacitracin. What alpha hemolytic groups are going to be sensitive to are optoken. Okay. So good things to know, potential quiz or potential exam or potential homework questions, compare and contrast beta hemolytic bacteria. And there are different groups, B and C, and then group D for the alpha hemolytic. Okay. Different species that represent beta hemolytic.
All right, welcome back.
Streptococcus pyogenes.
Good.
Oh, someone I think unmute.
On Wednesday last week. Oh, nice. Where'd you go? I went up to Seattle. Got a few friends living up there, and kind of
Right.
a little road trip before I actually
Mute him.
go to work. Yeah.
There we go. It's Brian. Okay. I'll have to deal with Brian later. Streptococcus alligactiae and then Streptococcus pneumoniae. These are the some of the more prevalent ones. Beta hemolytic, these are going to be the more serious pathogens in streptococcal variety because they have more complete hemolysis. They're going to be found in the throat, the nasopharynx, sometimes on the skin, usually more internal though, on the mucosal membranes. Lots of virulence factors, okay, for pyogenes. And you should be familiar with these. So they're going to have, that's a little cut off there, but this thing called C carbohydrates. So these are all going to be on the surface. These are going to be things that are potential antigens. So things that our immune system will target to then make antibodies to attack the subsequent invaders that have these same antigens. And some of those are the C carbohydrates, which protect against lysozymes. So remember, we talked about how different bacteria have strategies to evade once they've been in, so some can invade phagocytosis altogether. Some can be phagocytosed, but not be broken down once they're inside the macrophage. And that would be the use of this C carbohydrate. It prevents the lysozyme from digesting it. Fimbriae, we just know that those are for attachment. The M protein is what's going to resist phagocytosis. So not only does the Pyogenes strain resist phagocytosis altogether, if it is phagocytosed by a macrophage, it can then subsequently say, oh, okay, well, you got me. Well, you didn't get me completely because now I'm going to evade your lysozyme degradation. Hyaluronic acid is going to kind of allow it to go under the radar of the immune response altogether. And then the C5a protease. What's a protease? It breaks down protein. It's going to combat the neutrophil response. Okay. And so this is just a cartoon of where all these different antigens on the surface are located on the cell. I'm not really concerned with you being able to diagram this. It's just to give you a visual representation, but it would be good to know these different virulence factors and what their functions are. And that's what makes Pyogenes such a potentially vicious bacteria, because it has all these different ways to evade not just one type of immune cell, but a whole variety. All the more better that they're more susceptible to heat and disinfectants and desiccation. So those are the surface antigens. There's also toxins that they release. The streptolysins, which we said was involved in beta hemolysis. It also leads to necrosis. So it breaks down the cells. And then when you break down the cells, you damage the tissue that those cells make up. Erythrogenic toxin is pyrogenic. What's a pyrogen? Ooh, going back a few weeks. What's a pyrogen?
Isn't it something that can live in extreme temperatures?
No, that is an extremophile. It's right here. It induces fever. Okay, so a pyrogen is something. Now, pyro, I see where you were getting that, because pyro, hot, right? Fire. But yeah, so a pyrogen, this is what is the immune, this is what, not the immune, this is what the body is going to use as what line of defense is a pyrogen? Fever, in other words. Oh, man, I'm making you guys.
Is that a second line of defense?
Yeah, so right, you got to have this. You have a fever before your immune system kicks in. That is initiated by detecting these toxins. Super antigens are what are going to initiate monocytes and lymphocytes and lead to the release of tissue necrosis factors, so TNF alpha, TNF beta. And then there are extracellular enzymes such as streptokinase, which digests the fibrin clots. Uh-oh, man, just when I said that we can wall off these infections, preventing them from being systemic, now the bacteria has something that can digest that wall, and now it can become systemic. Or hyaluronidase breaks down connective tissue. Hyaluronic acid is a major component of things like cartilage and tendons and stuff. And so hyaluronidase breaks down hyaluronic acid, and so breaks down your connective tissue. DNase hydrolyzes DNA. So essentially can break down your DNA. And that can break down the DNA of your tissue cells. It can be a defense against the immune cells. If you are being engulfed or you're being attacked, if you can secrete something that is going to go into that immune cell and then start destroying its DNA, it can't now make certain proteins that it needs to attack you. And so, it's this warfare between the host and the microbes. Epidemiology, pathogenesis of Pyogenes. Humans are the only reservoir. So we don't get this from other animals. We get this from other humans by contacting, physical contact, by droplets, right? Respiratory droplets, or blood droplets, right? Contaminated food and fomites, right? Doorknobs and stuff. Which is why washing your hands is so important, which is why, not touching doorknobs in peak illness season, like right now, is important. Children are the ones that are most affected by this kind of infection because they're the ones that are licking the doorknobs, right? They're the ones that are touching things and then, sticking their finger in their nose or their mouth. So they're much more susceptible. One, because their immune system isn't as developed. Two, because of their behavior. Luckily, it's treatable and they usually have a sore throat and then they get over it. But if it becomes systemic, this can lead to progressive sequelae, which, remember, sequelae, prolonged illness, damage from prolonged chronic illness that goes untreated. Impetigo, as I mentioned, is going to be predominantly in children and infants, very contagious. So if a child has impetigo and they're interacting with other children, chances are those other children are going to get impetigo. Erysipelas is going to be something that is, again, coming through injured skin, which is your first line of defense, right? And then spreads throughout your upper skin layer, but then can go subcutaneously later on to become systemic. Okay. Let's see. I'm going to stop there because it's 10:53. How far did we make it? Yeah, not as far as I would have liked, but that's okay. We have Wednesday. Any questions on that? Any questions in general?
Actually, I do have a question. So we just talked about having like the hyaluronic acid capsule. Does it also, like, I know hyaluronic acid is also a humectant. So does it also help retain moisture?
Hyaluronic acid, does it help retain moisture?
Yeah, like in the capsid, like the hyaluronic capsid.
Capsid. The capsid? What do you mean by capsid?
Or hold on. Like I must miswrote it down. It's one of the virulent factors.
Hyaluronidase.
Capsule. Yeah. Well, it said hyaluronic acid capsule provokes no immune response.
Yes.
So with that capsule, I know, like I as I mentioned, hyaluronic acid is a humectant. So I was wondering if that helps keep that.
Yeah.
Keeps it moisturized or hydrated.
Yeah, exactly. So precisely because capsules are, that's the intent of the capsule, primarily is to prevent desiccation. So yeah, see, it's all interconnected, right? It all makes sense if you can, link the different pieces. It does make sense. But yeah, very good point, Hava. Thank you. Anything else, guys? Okay, so no lab discussion today. Instead, if you got a message from me, I sent a time that I want to meet with you during the next couple hours to discuss the research proposal. If you didn't get a message from me, make sure and just check your Canvas message inbox. And if there's no message there, you're done for the day. We'll see you on Wednesday. Your kits should be able to be picked up after lecture. And we'll go from there, okay? I'll try to get the homework assignment posted soon. And I just got a comment. thank you. The science club meeting, there's another science club meeting today at 1. So if you're around and you'd like to attend, it's my Zoom link. So just come back onto this Zoom and we'll talk science stuff. Okay. Okay. Well, that's it for today. And I'll be talking to some of you in just a little bit.